means and how we manage actually to develop some of these activities in our institution. So this is me, and the very first thing that I want to highlight is the collaborative nature of most of the things that we do. I mean, this is a very complex field, and I can tell you that you cannot do it alone. Okay, you can do it, but it could take a very long time. So I strongly recommend to collaborate, to participate in networks, in scientific societies or professional networks, and also, if it's possible, to create your own network if that doesn't exist. This is the reason why I'm citing here some of them, and I will give you some examples later on. I also wanted to highlight that although I am employed by the Blood and Tissue Bank in Barcelona, my research laboratory is actually a hybrid laboratory between the Blood and Tissue Bank and Heidelberg Hospital, and I'm also a researcher at the university where I coordinate a master's program. I wanted to introduce a little bit what is in Spain and also in Europe a Blood and Tissue Bank, because we are a public agency, so it's a non-profit institution of the Catalan Department of Health, and our mission is to guarantee the supply of blood and tissues. It's what we call the SOHOs, the substance of human origin, and also we are a leading center in immunodiagnostics. This allows us to innovate in SOHO-based therapies, and as you can imagine also in the ATMP development. And everything starts from a donation. It doesn't really matter we're talking about the same patient or from eligible donor, but these figures here that come from the Catalan Agency for Transplantation, I think they're very revealing, because they are telling us that from one ideal donor, this doesn't exist, but from one ideal donor, we can recycle up to 8 organs and 200 tissues. So you see the huge potential of these donations in order to improve the quality of life of patients, and in some cases to save their lives. And how we can transform those donations into personalized medicines. And starting from the structure of the blood, cell, and tissue establishments, that I'll give you as an example, the Blood and Tissue Bank in Barcelona, and this is quite common in Europe. We have a unique organizational model with a specialized workforce, dedicated equipment and facilities, core services that can help us with, for instance, the diagnostics, the safety, and this is important because it's also integrated with the national health system. And of course we comply with the strict ethics, regulatory, and quality compliance. This allows us to have these different lines of activity, diagnostics and safety, the SOHO donation and processing, and that is helping in order to develop these innovative therapies. Innovative therapies that not always are ATMPs. It could be the SOHOs, new users. They could be minimally manipulated, but also substantially manipulated as an ATMP. And you see that we are also interested in several products, like extracellular vesicles, for instance, or purified proteins from these donations. We can use them alone or in combination. We can make it as complex as we need in order to meet the requirements of our patients. And giving here as an example, the use of new 3D bioprinting technologies. But when we process these SOHOs substantially, then is when ATMPs enter into the equation. And this is the definition from the EMA, from the European Medicines Agency. And I want to highlight this second sentence. This is the promise of these advanced therapies. They offer groundbreaking new opportunities for the treatment of disease and injury. And this is the text that was written by the regulators. So you see that back in 2007, when this text was approved, they anticipated, a hope for the patients. We will see in the next slide whether we actually met that hope or not. And one of the things that I want to highlight here is the importance of having a specialized workforce. In the context of the ICT, we've been working in identifying the different segments of the value chain, from the donation to the treatment and also the follow-up of the patients. And what is exactly required at each of the different segments. Because it's likely that the professional who is working here is a different professional working here with different skills, different expertise, different competences. So we published that a couple of years ago, and this was a work done at the European Executive Committee of the ISCT, and it took into consideration, the scientific progress, technical progress, and it considers also everything that has to do with the risk assessment, with the medical needs of the patients, the costs, and of course regulatory quality and ethics. How to do that in a way that is sustainable? How can we meet all these requirements for the training of these people? In my case, I coordinate a network that is called VTCs for ATMP. We receive public funding from the European Union, and the idea is to create this European network of establishments that process substance of human origin, the SOHOs, but also all the relevant stakeholders. So this is not limited to blood and tissue establishments. So we try to convince and engage all the different stakeholders with expertise in cell biology, immunology, bioprocessing, with the aim of harmonizing all the procedures and criteria on the quality of the ATMPs. In the previous talk, Professor Weiss talked about the importance of producing quality ATMPs. And that's so important because in the future, you perhaps will perform a systematic review of the literature, of the clinical trials that have been conducted, and a meta-analysis. But how can we analyze that data if we don't know what the cells are? We really need to define quality control methodology. We need standards in order to make sure that the cells that were used in Thailand or the cells that were used in Spain actually are comparable. So this is so important that we decided to create this network, and actually we have now more than 100 members from all different European countries. But that is the, let's say, the overarching structure. Each individual country in Europe have their own network of advanced therapies. In Spain, we have TerapPlus. TerapPlus is a Spanish network. We try to connect researchers, clinicians, manufacturing platforms. And it's important to mention here that in this specific case, industry is not allowed to participate in the network. That is not the case for the BTCs for ATMP. This is a truly open, public and industry consortium. Here is only for academic centers, but fostering the collaboration and the decentralized manufacturing of ATMPs. The goal is always the same, to accelerate patient access to emerging therapies across Spain. How to do that, considering that ATMPs are not a classical drug? Probably this is obvious, but I wanted to show you these slides. Clearly, the nature and the composition of aspirin or an antibody is completely different in terms of size, complexity, the diversity of the donation to a living cell, because we're using cells that are alive, and because they are alive, they can respond to the environment and secrete whatever bioactive molecules that are needed. But those cells are coming from donors, and those donors actually are any of us, and we are diverse. So we could have a different HLA, different ABO group, we have a different clinical history. So all that counts when we are using cells in this cell therapy. So that is something very important to bear in mind. As I said, the regulation in Europe is from 2007, so it's very old, almost 20 years. But on December last year, the new regulation, the text of the new regulation has been approved. We are waiting for the final approval by the European Parliament. That will happen sometime this year, but it's not yet there. So today what I will try to do is to show you some of the points of the current regulation and perhaps some of the challenges that the new regulation are trying to sort out. In any case, please, I'm referring to this, to the quality. We need to demonstrate the quality, the safety, and the efficacy. It doesn't really matter whether you are a non-profit institution or you're a startup company. This applies to everybody. This is a picture from one of our labs in the GMP facilities in Barcelona, and we have a specific GMP for advanced therapies. So the regulators understood that the nature of these therapies is different from small molecules, as I showed you. So they created this specific GMP that takes into account the peculiarities of the manipulating cells and tissues. And I really like this slide. It is very simple, but it shows you the different milestones that need to be accomplished along the process of the development of ATMPs. Now we know that the starting material is composed of cells and tissues, and it requires the authorization by the agencies for transplantation. It's not only the cell and tissue, it's also the hospital where the tissues will be extracted. Our regulators inspect the hospitals and they say, okay, you can extract a bone marrow from this specific hospital, but not from another one because they don't have the SOPs in place, for instance. And of course, we need to get the approval by the ethics committee. Then there is the preclinical stage where we have to comply, of course, if we are performing safety studies, the GLPs, and then once you start with the clinical trials, as it's been explained before, the GMPs, the GCPs, the GDPs for the distribution of the cells, and then we have different different pathways in order to reach the patients, and I will go back to that later. I also wanted to mention other quality management systems that are relevant here. So it's not only about GMPs and the GLPs and the GDPs, it's also the value chain from the very beginning, from the donor to the follow-up of the patients. And in the case of the IST, we actually participated in the creation of a quality management system that is called JC, originally for hematopoietic stem cell transplantation, but nowadays the CAR-Ts actually use the JC for the leukapheresis and also for the infusion of the CAR-Ts to the patient. So you see how the quality management ecosystem is important. Specifically for the blood and tissue bank in Barcelona, we have this quality management ecosystem. So we have the compliance with some generic quality system, like the different ISOs, but also with a specific quality management system for the cord blood, the hematopoietic cells from the bone marrow, and as you see, back to the GMPs, GLPs, the EFI for the immune diagnostics, and the JC that I mentioned earlier. All this gives safe, allow us to process and demonstrate the safety of these therapies. However, when I show you the definition of the EMA, of the ATMPs, you remember that I mentioned the groundbreaking opportunities. So I'm afraid that there is a lot of hope, but at the same time this is a failure story. Because so far, and this data is very recent, this is from May 2025, there were 29 ATMPs authorized in Europe. 8 of them were withdrawn, basically because they were not reimbursed by the national health systems in Europe. The story is actually they were withdrawn because they were not efficacious. But if we go to the specific case of Spain, the patient access is very limited. From the 29 authorized products, look, only 5 are reimbursed by the public health system. And actually, in December 2024, Alofisel for the Crohn's disease withdraw. So this means that as of today, only 4 out of 29 ATMPs are available for the Spanish patients. So this is very frustrating because these patients can access Google and they realize that there is a medicine perhaps that could cure them, but that's not available. So they need to go to another country in order to get treated. We are lucky that regulatory authorities are helping in order to make possible to give access to patients to some of these treatments. The very first thing that we need to take into account is the possibility of scientific advice. And that's something that sometimes it comes too late in the development program. But you should be aware that that should be done at any stage of the development. And I really recommend you to do it as soon as possible. Of course, it's voluntary, but it will give you a lot of information on whether your development makes sense and you are on the right track. The questions could be on quality, non-clinical, clinical development. The questions must be specific. And it's important because you get a waiver if you are in an academic or a non-profit institution. but of course it's not binding, because the scientific and the technical progress, progresses every day, so it cannot be binding. Also, there is a specific support during development. Professor Weiss mentioned the orphan drug designation. We also have the PRIME, the priority medicines in Europe, but that's general for any medicine. We have something specific for the ATMPs. It's what is called the ATMP pilot. And today there are 5 ATMP developments, and one of them is from Barcelona, it's a CAR-T. And the idea is to provide extra support to a non-profit developers in order to make sure that these developments actually reach the marketing authorization. An important point is the hospital exemption. In Europe, we don't have a conditional authorization, but we have the possibility that if you have already completed a phase 1-2 clinical trial demonstrating the safety of your development, it is possible that for that specific hospital where the clinical trial was conducted, to get a conditional exemption. And then you can treat patients, perhaps 30 patients a year. That's something that needs to be agreed with the regulatory authorities. It needs to be prepared on a non-routine basis. So it's not something to be in an industrial setting. It's important, it needs to comply with the quality standards. So this doesn't mean that there is a low standard, no. Actually it needs to meet the GMPs, the GDPs, and everything that is required for an approved ATMP. So you see, there are many authorizations that you require along the process. The main problem in Europe is that there are so many different countries, and despite of having like an overarching directive, a regulation that applies to all the different countries, this specific exemption is transcribed differently in each member state. In Spain, as of today, we have 5 hospital exemptions. So this means 4 authorized ATMP by EMA that are reimbursed, and also 5 ATMPs that mostly are 2 CAR-Ts, and then we have MSD for spinal cord injury, chondrocytes for arthritis, and also skin for burns. Again, this is only for the specific hospitals. So if you are in one region of Spain, and then you need to be treated with the CAR-Ts, then you will go to Barcelona for the CAR-T treatment. But at least this is covered by the Ministry of Health. And here, I wanted to stop just to mention Ariadna. Ariadna was a patient, and she was very active in crowdfunding. And I think that the story is really nice, at the same time it's very sad, but I think that it gives us the hope that patients have a lot of power. So I also encourage that in the Thai ecosystem, to allow the patients to participate. The creation of patient associations and patient advocacy, because they have a say in all this process. And particularly for the CAR-T, actually we call it CAR-T ARI, they raised a lot of money that was crucial in order to develop the CAR-T ARI. So that was the first part of my talk, was very general, just giving you some of the learnings from the regulatory, the quality, the manufacturing. And now in my second part, I wanted to go specifically to my experience at the Blood and Tissue Bank. I will also talk about MSCs. You know now a lot of things from previous talk and also from your experience at Chula University. I work with MSCs from different origins, from the umbilical cord, from the bone marrow, and also from pluripotent stem cells. And we developed a panel for the QC of these cells. We try to make it as comprehensive as possible to define the identity, the purity, the potency of these cells. And also we try to understand whether the design of our bioprocess is yielding cells that are very compatible from batch to batch, no matter whether the donor is a different donor. And actually I think that we did that. These are 3 different donors from bone marrow, 3 different donors from the umbilical cord, 3 different donors from induced pluripotent stem cells. You see that they are different actually. So depending on the origin, they're not exactly the same, although perhaps the potency is compatible. But at least we managed to make sure that from 3 different donors of the same tissue, we get a product that is compatible. And we really spent a lot of time and effort to make sure that the critical quality attributes of these different MSCs is consistent. And then we have different categories of critical quality attributes. I mentioned the identity, the purity, and the potency, but of course in GMP we pay a lot of attention to the safety. How we did it, of course following the guidelines from the European Medicines Agency, but also, and that's very important, and here you have an opportunity, is also to pay attention to consensus papers. And this is something that is done by international societies. And in the case of the IACT, where I belong, there are many white papers, there are many recommendations. We give you some advice on what can be done. Of course, you have to apply specifically for your case, but it's good for guidance. And then to recognize the work of pioneers, Titanics, Takeda in our case. I mean, they did things that are so great like this, like defining what we call the basic preclinical package. So what should we do in vitro, what animal models to demonstrate the toxicology, the tumorigenicity. So this, of course, needs to be applied to the specific indication of the medicine that you are developing. But I think that it's quite inspiring, and then you can adapt to your reality. Okay, and what is your case? Oh, your case is different. Because if you ask me, the best animal model is the patient, but that's not ethically correct. So we need to use in vitro models, we need to use animal models, and of course I cannot answer your question because it really depends on your product and your condition. But you need to pay special attention to the level of evidence of any of these models is given to you, and also the relevance to the pathology. Because we can inject cells to mice, but, well, perhaps the results could be misleading, so careful with that. One specific example of one of the developments in our lab, this was the very first development. So now, looking back, we were very naive at the time, but with a lot of energy, we actually prepared the test ATMPs in compliance with all the quality and the regulatory and ethics. We applied the good laboratory practices for the preclinical studies, the good manufacturing practice, the GCPs, and you see how we start with a proof of concept in a translational animal model, then we perform biodistribution studies, toxicity and efficacy, and then we managed to get some money. All this was non-profit. I mean, we got money from the Ministry of Health, we got money from charities. It was conducted by collaborators at the hospital, PhD candidates. So all this took us 7 years and a lot of effort. But we were very happy to complete it. You see that we published several papers, so all the information is summarized there. The experience was good, but we learned a lot. We learned about the design of the clinical trials. We had money only for 15 patients. Then we learned about the potency, the power of the clinical trials, and then we did better in the new ones. I'm going to give you a couple of examples of those new clinical trials. We have done a lot of work in tissue engineering. I really believe that one of the true applications of MSC are to modulate the immune system, so we use them for GVHD, and I will show you later, and another one is for tissue engineering. So we've done a lot of work in regenerating bone. So we use bone particles from our colleagues from the tissue bank, and then we can create these 3D structures, living structures of MSC progenitors. And here I'm showing you some examples that the cells actually are happy on the surface of these scaffolds. We can actually model manually or using 3D bioprinting, and actually we use them in different animal models to demonstrate again the safety and efficacy of this. We use them in 3 major indications. One of them is femoral head osteonecrosis. The paper is about to be published, so I'm showing you only one example of a patient where with osteonecrosis. In the randomization, he had one femoral head receiving the treatment and another one just with a decompression. And you see with this imaging methodology that we can see metabolic activity only on the leg that has been treated with the cells. I cannot show you more now, but hopefully the paper will be out soon. I can show you a little bit more on this other one with the cell arthrosis. So you have to imagine a fracture of a long one that is not joining again. So we need to add some biology there in order to help it to join. So actually here we have done some work with the cells, with these structures, but also trying to identify biomarkers that are predictive of consolidation of the bone. And here, again, the number of patients is quite small, but in the next one, with 66 patients, I'm quite proud of that, we could demonstrate actually efficacy of the treatment. And this efficacy, that's very interesting. We were comparing this with the gold standard. The gold standard is an autograft in the fusion of vertebra. And we saw that the amount and quality of bone was comparable at 6 months with the treatment ATMP compared to 12 months of the autograft. So it was faster and it was better for the patients. So this is the example that we have now close to the hospital exemption. This is something that hopefully in the future you will see listed as the 6th or perhaps more hospital exemption in Spain. This is another example of a spinal cord injury, so collaborating with the surgeons, and you see again the development program in compliance with the different standards, GMPs, GLPs, studies in vitro, studies in vivo, 2 clinical trials. So I'm not going to go to the examples, but I have a movie that perhaps you would like to see. If I manage to do it, I believe it's this one. Oh, perhaps not this one. It's a movie about a clinical trial with myocardial infarction. So we had the possibility to work with a cardiologist and with patients that were candidates for a coronary bypass. So what we did here is a first-in-man clinical trial using a bioimplant. So there's a tissue engineering product composed of pericardial tissue that came from the tissue bank. And then here you see Christina, in our lab. So she was bringing the bioimplant to the hospital. That is in compliance with the GMP. Perhaps we are not as fancy as Novartis. And this is an open heart surgery. So here there is the location of the infarction, here the cardiologist. So she's now taking the tissue engineering product that is composed of pericardial matrix and the cells. Here you see that the patient is on an ECMO, and now she is using the cells are only on one side. So now the side with the cells is touching the myocardial infarction area, and now she's going to fix that with surgical glue. We were very scared at this point, so perhaps at any moment that the heart is beating, then the bioimplant would just be ejected, but that was not the case for none of the patients. So this was quite revolutionary at the time. It was published in 2020, the first paper, 2023, the second paper. We treated only 15 patients because that was a first in man, and we didn't see any toxic effect of this. We saw actually like a systemic effect on the inflammation. And again, because the power of this clinical trial was quite small, we couldn't see more information on the efficacy. However, please pay attention to the administration. That's too harsh. One of the things that we realized that this will never be an hospital exemption because the feasibility is very complex. So now we're moving to other strategies like using extracellular vesicles, trying to use alternatives in order to avoid, open heart surgery. Another thing that is a concern is, okay, if we are not using cells from the same patient and we are using donor cells, allogeneic origin, then we need to pay attention to the compatibility. I know that in many times we are told that the compatibility is not required, but in my experience, there is some requirement. Or if we try to match them, perhaps we will have better outcomes. I cannot demonstrate that, but we are going to do it in the coming years. And it's true that we have a lot of information from hematopoietic stem cell transplantation. Perhaps there's not that much information from different tissues. This is a paper that we have published recently from one of our PhD candidates. And actually, most of this information perhaps is not true because it's masked by the use of immunosuppressors. So we try to understand this better, and that will help us a lot for the new generation of advanced therapies. This is a small diagram of the interest in my lab. So we have some applications of the MSCs in the autologous context. We have some applications of the MSCs in the allogeneic context using MSCs from the umbilical cord. And now, currently, addressing these compatibility, we are using MSCs from pluripotent stem cells and also the extracellular vesicles. Why pluripotent stem cells? Probably you know very well this. We have now the technology in order to, thanks to Professor Shinya Yamanaka, to reprogram whatever cell into a pluripotent state. So it's possible to reprogram umbilical cord blood from very specific donors with HLAs that are very compatible, in our case with the Spanish population, to make them into a pluripotent state phenotype. And you see the cord blood of the donors has been selected by the immunobiologists in our lab, to make sure that they're very compatible. So with here it says that with in our case with seven cell lines, we can cover 23% of the Spanish population. But if you allow one mismatch, we are covering more than 40% of the Spanish population. And now from those IPS, you can make any cell type. Your favorite cell type, can be derived from the IPS. In my case, I have interest with for the MSCs, but some people at the proton tissue bank is interested with NK, with T cells, with macrophages. Now, we will be able to, to generate again the same products that I showed you earlier, but with more compatibility with the patient. And here, you see Maribel and Laura working with bioreactors, with Tiny Prodigy, because if we are able to expand very compatible cells, we're going to do it, mass production of these cells in order to cryopreserve them and make them ready off the shelf. However, we cannot do it manually because this will add variability, it's complex, there is a risk of contaminations. So that is the reason why we have interest in using bioreactors. However, the question is, if you are producing the cells manually or if you are producing the cells in bioreactors, are those cells compatible? Are they the same? We mentioned about the differences between different origins, but if the manufacturing process is different, are the MSC also different? We have some regulation there. The regulators actually give us some advice on how to conduct the compatibility studies. And this is something we're doing in the lab, because you don't want to do it when the new development is very much advanced. Otherwise, the regulators will ask you to repeat the clinical trials. And as I mentioned, we are non-profit, we are poor, so that would be a big problem. So, and also another thing that I want to mention is that sometimes we don't know much about the mechanism of action. So this adds some complexity in the compatibility studies. But this is a topic that in the context of the ICT, in the context of international collaboration, we should address. I don't have an answer today for this, but I have some ideas. And the idea is a paper that we published just last week. I was lucky that I was in Madrid for the EBMT meeting, and we had an EBMT ISCT joint session, and we talked about potency, the potency testing. And from the initiative that I coordinate in Europe, from the BTCs for ATMP, we have just published this paper in which, according the stage of the development of your ATMP, according to the TRL that we heard about today, you could actually start with a different potency test, and then you can make it from qualitative to semi-quantitative to quantitative, and then end up with something that the regulators will like for your final product. But careful, we know from Mesoblast, we know from the Ryan Seal, they had lots of problems in order to get authorization from the US. And the reason is because we don't know what is the mechanism of action. And sometimes we are creating a very detailed artifact in the lab that has nothing to do with the mechanism of action in your patient. So careful about that, because you could invest a lot of money, a lot of time on something that, again, name insists, is an artifact that has nothing to do with the reality. Here you have the reference of the paper if you are interested. So some final remarks from my talk is that Of course, ATMPs present many challenges that are beyond the scientific. I mean, the regulation, the technology, that is something that we need to understand. But I think that it goes in parallel with the regulations. So if we can do it in a way that we team up with the regulators, don't see the regulators as your enemy. Actually, it should be a facilitator for you not to waste time and then to address specifically the points that are important in order to make ATMP safe, efficacious, and feasible for the treatment of the patients. And another thing that I think is quite sad is that regulatory success is not guaranteed for access for the patients. And you saw that from 29 authorized ATMPs in Europe, in Spain, we have only 4. So that is the reason why the regulators also try to find other pathways that can facilitate this access to the patients, and that is why the hospital exemption exists. Finally, this is visual, but I think it makes a lot of sense. We have this triangle. We care about the quality of the product, something that is very much related to the treatment of the patient, but that needs also a donation. So we need to make sure that we understand the eligibility criteria for the election of those donors. This is what we call the vein-to-vein model. The vein-to-vein model is very old. We use it for blood transfusion, we use it for tissue transplantation, and now we are applying the same concepts for the ATMP development. So before I conclude, I wanted to thank people in my lab. These are the different lines of research, as I said, for the different treatment. I didn't give examples today for the extracellular vesicles. We have also some engineers because we develop bioprocesses using bioreactors. And I didn't mention today everything that has to do with our projects on bioethics and teaching, and also some cooperation programs. I have a very interesting cooperation program with India and Sri Lanka on a training for transfusion medicine. So if you are interested in this, I recommend you to watch this video. It was a very nice experience in a TEDx talk. And I really thank you for this opportunity to tell you about our experience.